Hey folks, all right, good to see you all. Welcome back. A few quick announcements. More of this will be coming out on Piazza as well. Rishi Anjali will post some stuff about this and just enable transcript. Okay. The first is that update about lecture. So as you know, I've been looking forward to seeing you folks in person in lecture. And it's been some weeks now since campus has transitioned to that, and I haven't been able to do that, unfortunately, because of my health. And unfortunately, I have a bit more clarity about that now. And it looks like because it's going to continue to be that way for me, unfortunately, for the rest of the semester. So unfortunately, I'm going to need to be staying at home and continue to do Zoom lectures for the foreseeable future. So I'm sorry about that. The staff are going to try to take another pass at the technology problems that we encountered before in trying to set up a live, an in-person option for in the lecture hall, even though I won't be able to join physically. So they're planning to do some more testing and some sort of pilot tests about that. So watch out for some info about that in the near future. So that's thing number one. Thing number two has to do with the exam. We have some more thinking to do about this and to figure exactly what we're going to do about it. But the long and short of it is a couple of things. Number one, thanks for all your hard work in preparing for and taking the exam. The exam was a bit of a miss, and this is really on my part on writing. It was just too long. And in part that was because of there were just too many programming questions, and the way that we logistically handled it through Gradescope with formatting created just too much time pressure this time. So the mean on the exam was lower than intended and out of line with what we are expecting and intending with regards to the grade bins. So we're going to correct that in one way or another, provide an opportunity to fix that. So we're working on some of the details on that. It might take us a bit more because there's some moving parts to all bring together. So just know that for now, and that we're working on it, and we'll update you when we have some of the ducks lined up, when we have all the ducks lined up in the near future. Okay, so those are the two announcements for today. Okay, let's get going with lecture for today. I'm really excited to share with you this week an introduction to color science. Actually, a lot of my research and in my research group has reoriented around study of color in visual perception and sort of the frontiers of what color perception can look like by direct stimulation of the retina, and considering what that means in sort of the limits of human visual perception. So this is a topic that's kind of intellectually really resonant for me, and something I've been spending a lot of time thinking about recently. Okay, so this week we have two lectures. We may even spill into next week, because at the beginning of next lecture, we are going to have some of the TAs come and talk, give some more demos to you folks and talk to you more about the final project. So I'm going to give that as much time as they would like to give it, and that you have questions for, and then we'll just sort of pick up and take as much time as we need this week, and maybe spilling into next week for this lecture on color science. But let's get going. And I saw Rishi joining, so I think the slides will be posted for this on the web very soon as well. Okay, so let's start with some basics. Why is color science? Why is color such an interesting topic? Well, it's for very basic human reasons, right? Color is really central to our human experience. We're highly visual animals as human beings. And so much of our experience in the world is visual as we move through the world. And it's like a bedrock quality of our visual perception is color. If you've ever seen a sunset in the natural world, it's one of the most striking things because of the brilliance and unusual quality of the color of the light at that time of the day. It's of universal fascination and beauty. As children, we're always talking about color. I have kids that are 6 and 8-year-olds now, and their whole lives, a huge fraction of the time, is consumed with thinking about color, talking about color, playing with color, drawing, experimenting with how to do different color combinations or how to mix different colors. And this is a core part of art, of course. This is one of my favorite paintings, is Color Study, Square with Concentric Circles by Kandinsky. Many artists' work is almost, entirely centered in color and explorations of color. Mark Rothko is one such artist that's well known for this. He has these large color panels, often in three rectangular panels or three rectangular sections, as you can see here. This one, number 61, Rust and Blue. And sitting in the presence of one of these kinds of paintings and its magnificent color is sometimes described by aficionados of Mark Rothko's work as almost like a religious experience. Of course, away from the artistic world, just in the natural world, things like the beauty of blossoms, of a rose, or of the exotic species of animals like peacocks. The brilliance of the color are things that are fascinating to us, and that are set up in part because this kind of coloration can be so attractive in mating rituals or drawing attention to themselves, either in species or across species. As humans, we use color, of course, also for, greatly for communication, for symbology. So almost universally around the world, fire engines or fire stations are red, right? Red because of its brilliance in attracting attention and making the vehicles or the buildings associated with this stand off to represent the importance of the urgency of their mission. Or symbology in other ways, like following a yellow brick road because of culture, from in this case, from the story of the Wizard of Oz. So color just plays this central role in so many different dimensions of the human experience, from every sort of waking moment of visually observing the world through to just a sprinkling of some of these examples that I've tried to set up. Oh, thanks. This is my eldest daughter when she was a bit younger. Along this vein, I just want to take a slight segue and share with you, if you haven't heard about it before, this technology, this company called Enchroma. I think this is a really interesting company, really interesting technology. It's spun out of, in part, out of some technology from Berkeley Vision Science. The idea at a high level is that if you have color vision deficiency or color blindness, for example, the most common version of color blindness is which affects over 5%, 5 to 8% of the population, is of course red and green color blindness. Then this company has a kind of glasses with a kind of filtering, which is a filtering in the spectral domain that has certain notches in the spectral, in what's wavelengths it passes preferentially. I don't have time to go into the details of how it's intended to work. And there's actually a bit of controversy about it in the vision science community. And they're sort of tweaking this formula all the time. But if you're interested, come to office hours. I'd be happy to chat about it. But at a high level, the idea is if you put these glasses on and you have one of these color vision deficiencies of red-green color blindness, then the idea is that it will enhance your ability to differentiate the kind of colors that usually you haven't been able to differentiate. Now, as I said, whether this works or not is really varies by subject and is a little bit controversial. But one of the interesting things this company did was to make some marketing videos where they said, "Hey, buy it for a loved one, and then when that person puts it on for the first time, just take a video, with your phone, and then send it to us, and we'll sort of compile these." Now, it's a marketing program, so you got to take it with a bit of a grain of salt. But having watched a lot of these, I want to play for you just a couple of these. This first one is my favorite by far. Here you go. So this is, if you will, a colorblind person's reaction to perceiving new colors for the first time.
Oh my God!
Your phone looks different?
It's like this looks pretty similar, but this pink, like this.
Yeah, it doesn't show new colors. That's right. At a high level, well, it'll take me a little bit more to give a technical explanation of it. We'll need some things that I'm going to describe later in this week's lecturing. Okay, so let me come back to that a bit later. But the first thing is like, just in this introduction, as I said, color is so central to our human experience, and but there's a significant fraction of us that don't perceive the same range of, or the same range of colors, the same color gamut. A gamut is a space of colors, the same space of or gamut of colors as quote unquote normal color vision. It's a significant fraction of the population. And I think these videos just sort of underscore some of the emotion involved in being able, or just the level of surprise in being able to perceive these colors for the first time, apparently. Okay, here's another one.
You can tell orange.
Yeah?
What else is it?
What are you talking about?
The streamers.
Wait a second. Ah! It's green. I thought it was all orange. How did I think it was all orange? That's green.
Okay, there's a lot of these out there. If you like them, they're actually like pretty heartwarming, some of these. There's a lot of crying in these. Actually, I didn't choose the most like weepy, but many people like, oh, they'll look at a sunset for the first time, or their kids' eyes and see their kids' blue eyes or green eyes for the first time. They'll just bawl. It's really quite striking. Okay, anyway, so I don't know exactly what this is telling me, but when I saw these videos, I was like, I'm not sure what is fact and what is sort of just sort of like interesting human reaction and great marketing, but it's teaching me something. And so this has been really interesting and sort of influential piece of thinking, at least in my research development myself. While we're on the topic of color vision deficiency, I wanted to just show you as well. If you haven't seen this kind of thing before, there's some sites that will try to simulate what color perception looks like. So let me give you an example. Here's this picture of those rows that I showed you earlier. So this is a simulation of what it might look like for a red-green colorblind person. Specifically in the subclass that this person would be called a protan. That just means, that's kind of technical. That means that this person has one of their 3 types of color-related cells on their retina. We're going to talk about the technical details of this later. One of those 3 types of cone cells, as they're called, specifically the L type of cone cell, is functionally deficient. Okay. This is a second one for someone called a deutan. This is the second type of cone cell called the M cone cell. The L and the M, it turns out, are functionally quite similar. So the 2 of these are functionally very similar to red. The most salient version of this is red-green color blindness, or inability to differentiate these in a normal fashion. This one's a bit different. This one is, or folks in this class, which is much more rare, would be classed as tritans. And this one is a lack of, or a functional deficiency in the S or short wavelength cone cell. Again, as we'll talk about those cells in detail later. Okay, so here's a sort of summary of it. So normally what it might look like in full color, or for a quote unquote normal color vision experience, and then for protans, deutans, and tritans, which basically have to do with functional loss or deficiency in one of the 3 types of cone cells. Yeah, someone sort of makes an observation that tritan and normal look pretty similar. It's true, they look more similar in this class because for tritans, there's sort of a red-blue axis that sort of persists, although yellow in particular is somewhat diminished. Now, you might ask, how do we know, that this is what it looks like for people? And what you really have to recognize, and this is, I think, one of the really interesting features of color, of course, that is so central to it, is that it's almost impossible to know what someone else's color experience is. We can sort of talk or transact through language to be like, okay, yeah, I'm looking at a red rose, I'm looking at a red apple, and sort of get a sense of, we can agree that that's red, and that redness is similar to the redness of a red rose and a red apple have a similar quality when we say it's red. But to know what it internally feels like, which is sometimes called the quale, the quale is the internal experience. Okay, to know what someone else's quale is really impossible. So how can we know what it is like for a colorblind person is a very natural question to ask. And this is really interesting. So I don't want to belabor this point, but just very briefly, and I won't even go into the graphs for this. If you're interested, come to Office Hours and I can tell you a bit more about this. They're really interesting stories. It turns out that it's very, very rare, but folks do exist that are colorblind in only one eye and have trichromatic or full color vision in the other eye. These folks are extremely rare. It's sort of a genetic property and a phenotypic expression that is quite unusual. But when this occurs, you can do, of course, tests where you show colors or say a wavelength of light to one eye, say the colorblind eye, only through some kind of very special setup that only puts the light into that eye, and then gets the person to match that color in a controllable fashion with the normal color eye. And from that, you can deduce certain things. For example, in this case, you find that for a person with a deuteranopic eye, which, as I mentioned before, was someone that has a functional deficiency in the M cone cell, that the person will map almost any wavelength of light, almost any color of the rainbow, to something that's either blue or yellow. So they sort of have a blue-yellow axis of color perception. Okay, and similarly for someone with a tritanopic eye, which is a bit different, that's the one that has a functional deficiency in the S cone cell. Okay, so just one last set of examples on this again. So this is full color. This is for protan again. This for deutan, and this one for tritan. So for the person that before in the comments was observing that tritan was a little more similar to full color. In this case, if you sort of put them side by side, you might sort of get a bit more of a sense of the color part of the color range that is deficient. In the full color, you see this very clear yellow, like in the apple here, or in some details of these pictures that's absent in the tritan. But similarly, certain features in the protan and deutan are present that are absent in the tritan. Okay. Anyway, okay. So that was a brief aside just to show you this exists. Okay, to try to simulate for full color vision people imagery that attempts to approximate what it's like for a colorblind person, but it's based on sort of this very specific sort of studies, and really it's not possible to know how accurate this is experientially. Okay, so that's sort of one set of things, one set of observations about color, how central it is for us as human beings. But now I want to talk a bit about some of the key characteristics of color that make it really rich to study and tricky to deal with scientifically and engineering wise. One of the really striking elements of it is something called chromatic adaptation. Okay, so what is chromatic adaptation? It's just that different objects look different under different lighting conditions. Okay, and yet, even though they may be colored by the illumination, maybe colored very strongly by the illumination under which we're viewing them, as human beings, we are really good at factoring that out and being clear about what the color of the objects is. This is just a picture of someone studying chromatic adaptation. This is a very famous person in color science, Professor Mark Fairchild at the Color Institute in Munsell Color Institute in RIT in Rochester. Anyway, one of the ways you can study chromatic adaptation, you can see he's looking at a white piece of paper with a couple of color chips on it under a certain kind of lighting. In a booth like this, you can change the illumination in a very controlled way and do scientific experiments of this kind. A whole range of studies can be done. Let me show you one really interesting example of this that I think underscores one of the things that's going on. So here's a picture apparently under normal sunlight kind of conditions. Okay, so it's a basket of fruit and some objects. There's a blue towel, as you can see. There's a yellow banana, a red apple. Now imagine you have a cyan filter. Okay, now we're just going to sort of Photoshop this on top, sort of blending it in, but this would be quite similar as if you colored the light cyan. It's a very sort of blue, a very blue light source onto this scene. And at first, this is sequence of slides was created by Professor Fairchild. Okay, I'm not sure if you can see what's happened in this picture yet. So take a look around, sort of see what's going on. So the cyan filter has been applied to this picture, but only in a portion of the picture. Can everyone see it where it's been applied? You noticed it yet? I'm going to go back and forth in a second. Yeah. Okay, can you see that? It's on the banana, okay? It's sort of, the photoshopping is imperfect. So if you look closer, you can kind of tell by the edge of it. Now, isn't this interesting? Okay, so if you look in this scene, this sort of looks now like there's a green banana, okay, in this basket of fruit. All right, everything else, still an orange, still a red apple, yellow lemons, but the banana that was yellow before, if you recall, now looks green. So let me just go back a couple of slides. So it's a yellow banana, but with this cyan filter now, perhaps it looks green to you. Now let's apply, though, the cyan filter in a different way. Rather than sort of colorizing this one object, let's just colorize the whole scene. Okay, you see what's happened now? So the cyan filter has just now been applied uniformly over the whole scene. And if you look at this scene and let your eye roam around it, just let yourself adapt, what your visual system is doing is the name for it, the perceptual process that's happening is called chromatic adaptation. Okay, so your eye is adjusting to this lighting or apparent lighting on this scene. And you'll observe that banana doesn't look green anymore. It looks yellow, okay, even though it's exactly the same coloration. It's exactly the same RGB pixel values in this picture as in the previous. The apple still looks red, the lemons look green, the blue of that cloth still looks blue, and the banana is now back to yellow, okay? So this is an example of what's called chromatic adaptation, our ability of our human visual system to factor out the illumination. This is really turns out to be important in building camera systems, for example. You may have heard of the technology, called automatic white balance, auto white balance. Sometimes it's called by the acronym AWB. Let me show you an example, okay, of what happens if you don't apply it. So here's a few photos, okay, that have been taken. If you look closely at these pictures, they've been taken in different lighting conditions. Okay, so one is out in the sun. This one's taken in very dark conditions. Basically, the lighting just comes from the candles on this cake. This is an indoor scene with like a halogen light bulbs as the only light sources in the background. This one's interesting. It's sort of inside a building under an arch. So there is sunlight. You can see that the sun is out, but because this part of the building is in the shade, the only light that's coming to it is from the sky. Okay, this is a pretty typical outdoor shade lighting situation. And if you look at it, you'll notice this kind of looks cool. It looks kind of bluish. This is a sunset that looks kind of overly red. Okay, so what auto white balance aims to do is to normalize the color in all of these, the color cast. By computationally applying something similar to the neural process of chromatic adaptation. So here it is, okay? So this is before, no white balance applied. This is after auto white balance is applied. And you'll notice, okay, so this looks warmer now, this picture. It looks less blue. This one looks neutral, where before it looked really yellow, very warm. In this picture here, it looks a lot more neutral. Before it's highly, colorized yellow, and now it looks more neutral. A little bit, auto white balance doesn't always have to completely eliminate all of the color cast of the light source. In this case, because it's a candle leaving some of that color in, can keep some of the ambiance of that scene. Okay, but you get the idea, okay, of what auto white balance is doing. Just as a final note, some of these pictures are still a little bit dark because they're overly bright from the light sources. So it would be typical sort of in photo image processing or something to apply some more sort of photo finishing to brighten up some of the shadows or things like that. Okay, so you could do that too. I describe that as auto white balance plus some light manual editing or something like that. Okay, but this is the heart of sort of chromatic adaptation in an algorithmic sense. And it's a very simple algorithm. Okay, so just to share briefly with you about this. Okay, so if you take every pixel in that one of those pictures that you want to white balance, and say that pixel had an input value like RGB, that was the raw input value of R prime, G prime, B prime. We want to output an RGB value that's auto white balanced. What is it? Okay, so auto white balance turns out to be, like almost the most simple arithmetically stupid algorithm you could come up with. You just divide it by like an RGB value for the white point in the scene. So what does that mean? Okay, so R prime W, G prime W, B prime W, these are just the RGB raw pixel values for a white object in the scene. So if I go back to this picture here, right, this dress is white, but the RGB value is going to be encoding something that's yellow, if that makes sense. This one, the white on the shirt, is going to be an RGB value that's encoding something that's bluer than typical. Okay, so that's what I mean by this RGB value here. And then to output, you just divide it. Okay, you take the raw pixel value, you normalize it by the white values, and that's it. So really, it's very simple to apply. Really, all of the difficulty is in sort of trying to analyze a picture, an arbitrary picture that you're given, to say what is a white object in the scene, which is not always an easy thing to do. But that's sort of the heart of what makes auto white balance algorithms tricky. Yeah, so the question is, how would a camera determine what would be white? What should be white? That's sort of a bit beyond the scope of this lecture today. So let me just sort of say there's a whole range of algorithms that have been designed for these over the years. And these days, they tend to be pretty sophisticated and machine learning based in order to try to identify known objects in the scene. It could be faces, as well as an estimate from learning of what the complexion of the person is in the scene, and use that in order to normalize and estimate what a white point could be. Or it could be the brightest pixels in the scene, which can often be a good mark for white, but again, within sort of a learning context. What happens to parts of the image is brighter than the chosen white point? Do we have to let the values go over 1.0? Yeah, that can occur, particularly if they're very, very shiny objects in the scene, for example, that reflect in a mirror sort of a sense, more light than like a white piece of paper might, which is very white, but scatters light in all directions, so may not be full energy transfer. So for those, they would saturate. That's exactly right. In the technical portion of today's lecture, which is going to be, or this week's lecture rather, which is going to be on color reproduction calculations, with respect to white balance, we're going to implicitly assume either that auto white balance has been applied, okay, according to sort of how I've summarized here, or that the viewing conditions of the color reproduction that we'll look at match the original scene. What do I mean by that? So we are going to try to reproduce color like, okay, I have something in the world that is a real object. I want to reproduce it on my color display as an example. I could try to print it out too, but the examples we'll look at will mostly center on like, on an electronic display with RGB pixels, for example. So if I reproduce it and then I'm viewing it in the same lighting condition, then the same kind of chromatic adaptation process in our human visual system will apply. Okay, so those are the assumptions that we'll make today to sort of simplify it. Okay, so that's a little bit of a chromatic adaptation. Let's keep going through this introductory sort of tour of some of the really interesting visual characteristics of and surprising visual characteristics of the human visual system. Okay, the next point is color perception is super adaptive. It really, you can't stop your visual system. You can't stop your eyes or your brain from interpreting the colors relative to their surroundings. And all kinds of surprising things happen when this occurs. Okay, so let's take a look at a couple of illusions that have been created over time in order to sort of really draw out how dramatic this process can be neurally. So if you haven't seen this illusion before, this is a really terrific one. Actually, yeah, the dress would be a good example. It has more to do with white balance. It's also a little bit more ambiguous exactly what had been going on in that one. So I actually don't have the dress as an example. Some of these are a little bit more scientifically worked out. But similar in flavor, I think, in sort of like how confusing they can be or surprising they can be. Okay, so take a look at this scene created by Professor Ted Adelson at MIT a long time ago. I really think this is a phenomenal example. So what do you see here? Some kind of checkerboard. It's got like some kind of object casting a shadow, like a green cylinder casting a shadow from an apparent light source that's off to the top right. Okay, you see that? So what I want you to do is look at the squares on the checkerboard labeled A and B, and take a look and see, make a determination for yourself if one is darker than the other. Okay, color-wise, is one darker than the other between A and B? And then what we'll do is we'll start chopping it off and sort of see what happens. Okay, so we'll just block this part of the picture so you can judge A and B as one darker than the other. Turns out we can block out this part of the picture too, including the cylinder. Okay, so now we just have this tiny little snippet of this scene, A and B is one darker than the other. We can chop this part off. We can chop this part off. Is between A and B is one darker? Does one seem darker to you? And then for this last step, so this part you can pay particularly close attention to. So then if we just sort of chop off this middle part here that completes sort of the wiping out of the context, the adapting context, okay, is like, this A and B does one seem darker than the other? It's a pretty remarkable illusion, just remarkable illusion, okay? So you rewind all the way back here, yeah, those RGB values between A and B are exactly the same in this scene. And sort of seems like, okay, maybe it's because of the total context of this scene. There's this object that's casting a shadow. So this light colored square here happens to be in shadow, but you know it's a white and black checkerboard. So, but if you take out this out, you take the context that the object casting a shadow, all you get is sort of the shadow itself, a very narrow context on it. And the more you chop it off, you're like, but now it's just like some bare little gradient between these two, these little, they're not even checkerboards anymore. These little snippets of polygons, right? And if you just get rid of that final little bit, then the illusion is completely gone. But it really takes complete destruction of the context for, what was the theme here again? It's about how adaptive our visual system is. Okay, how adaptive it is to the context of the scene that you're looking at, and you cannot turn it off, right, visually. Here's another famous example by it's called a Lotto cube by the inventor. And the trick in this picture is to look at the central square in the top face and the front face of this cube and sort of judge, do those two colors look different to you or do they look the same to you? Again, you can guess where this is going at this point. So if you just darken everything else in the picture, it's really remarkable. It's almost like you can't believe that in that blacking out process, like some filter wasn't applied or something to them, but they're exactly the same RGB value. So, the slides are posted online and the PDF, you can go take a look at yourself and use your favorite color picker and go check the RGB value of these. They're exactly the same. So what is happening here? Well, apparently, again, the context of this cube with the front being in shadow, right? It's pointed away from the light source, the top face being brilliantly illuminated, apparently creates a sense of our interpretation of the color. If you're like me, you would call this a brown color. And you would call this a light orange color. And this is one of the characteristics of the human visual system. Actually, brown is an interesting color. Brown is not a color that you can identify in isolation. It is a dependent color. It can only identify brown relative to a context of other colors that it's in its surrounding. Gray is similar in that respect as well. You can't term a color gray in isolation. It's only when it's relative to brighter white or dark or black or other colors of different tones in the scene that you can perceive that. Okay, so another example of that context. Here's another example. Perhaps you can guess where this is going, but it's still quite striking, or perhaps you don't believe it yet. Even really simple judgments, about lightness. There are two sets of chess pieces here, so you can try to look at the two queens and try to gauge the relative brightness of the two. But then we can just sort of move. Okay, we'll sort of cut out this this top queen here, and we can just translate it down. And yep, it's exactly the same pixel values. So the context, these examples are simple examples, but carefully designed by these inventors in order to show just how much a visual system is contextualizing the interpretation of color, even down to just things like brightness judgments based on the context of what we see. Dissecting this a bit, taking it away a bit from sort of some of the visual aesthetics of some of those previous examples. These are examples of something that's called simultaneous contrast and the surround effect in human vision systems research or human vision science. Here's a simple example. There's rows of gray squares. Each of those rows, the squares, in each of the rows, the squares are exactly the same brightness. But behind these rows of squares, there's a gradient, right? As you can see, it goes from light on the left-hand side through to dark on the right-hand side. And you could compare, for example, your judgment of the brightness of this square versus this one, or perhaps this one's more striking, the darkness of this square versus this one in the bottom, and observe that relative to the surrounding colors, it can really affect your judgment of the relative or the absolute brightness of these tints or of these shades. This one's even more powerful, I think, an example of surround effects. So let me walk you through what's going on here a little bit. You see this grading of yellow and blue, and it's shown in sort of two different spatial frequencies. It's shown here, and then the same exact picture here is shown again, but squashed horizontally to increase the spatial frequency, as we talked about in our frequencies and sampling lecture, increase the spatial frequency of the blue and yellow grading. The same gradings in the bottom, but then there are these squares. The squares of color in the top are red, and in the bottom are this cyan or green color, okay? And the squares are existing, as you can see, either on the yellow part of the grading or on the blue part of the grading. And they're repeated over here, but they're squashed, so they exist on a finer grading. Now, if you're like me, these squares don't look like they're the same color at all. Okay, they look red. To me, this looks like very dark red. This looks much more orange red. And over here, it looks far more orangey than this one. But you know what's going on here. This is exactly the same red color in terms of RGB value, okay, across all of these different red squares and red rectangles here. Similarly for the green or blue or cyan color, whatever you're going to call it in the bottom. Oh, that's interesting. Iman says that they all look exactly the same. They look like the same red to him. That's really interesting. So feel free to comment on this. Why is it more pronounced at higher frequency? Yeah, so this is a feature of this surround effect, is that the shift in the color that's a typical perceptual response or perceptual characteristic for most people is that that color shift increases with the spatial frequency of the surrounding color grading. In fact, I can show you this to you a little bit more. We can just copy paste this image and just shrink it down over here. Okay, so this, by shrinking it down, the spatial frequency went up even more. And you can compare, for example, the red square or rectangles over here compared to over here. It's exactly the same image. And all I did was I used this presentation software just to scale it down. Okay, so there's no color shifting going on, or color change in the RGB value. It's just, it's purely a perceptual effect. All right. Top looks more different, the red to some folks. Squash ones actually look a slightly different color from the not squashed ones. Yeah, they do for me too. So the squashed ones definitely look to me like the color shift is more, is stronger. These look even darker red to me, and these look even more orange red to me, as an example. Okay, so this is an example of a visual phenomenon, a characteristic of the human visual system called the surround effect, or related to the broad class of these kinds of effects called surround effects. Okay, so that's just a little bit of fun stuff on the human visual system. Is highly adaptive, and if you're thinking about trying to make color judgments in complex viewing environments, you need to be pretty careful about it and cognizant of that kind of effect. Of course, you could imagine that you might be able to use that, and people totally do use this deliberately. Artists use it in graphic design or marketing. People use this in order to accentuate certain colors and draw things to present them more visually prominently, using or exploiting those visual characteristics that I showed before. Okay, now I want to show you a different aspect of the human visual system that we'll come back to later when we talk about file format color encodings. And this is something called generally, a color opponent theory, or the color opponent characteristics of the human visual system. And we'll look at it through some illusions that have to do with after images. The high level point is that some of these illusions are intended to illustrate the fact that, to you natively, that visual perception operates along color axes that have an opponent quality to them. They're sort of antagonizing colors. And you can draw out those antagonistic qualities by sort of taking a look at what happens when you stare at colors for a certain duration of time, and then turn them off and see what kind of opponent colors come to your vision. Okay, so these are after image illusions. Let's start with this one. So if you will, you can just sort of stare at this gray dot at the center of the screen, but really try to gaze at it and don't allow your gaze to move. I'll just describe a few things while you do this. It takes a little while, maybe 15, 20 seconds for this effect to sort of, affect your retina. Something, biophysical is happening, and then it will sort of radiate through the human visual system. So as you stare at that gray dot without moving your eyes, you can sort of notice at the same time, again, without moving your eyes, that the triangle at the top is yellow. The triangle at the bottom is this dark blue. Triangle off to the right-hand side, top there is red, and on the top left is green, things like that, okay? Now, as you stare at it, I'm gonna turn us to a white background. So keep staring for these last few seconds. And as I turn it to the white background, try not to move your eyes, just let it rest on the white background and try to keep it fixed for a few seconds at least until perhaps you perceive an afterimage. Okay, so here we go. We'll switch now. It may fade for you initially, but if you keep sort of staring, perhaps an afterimage may come back with some clarity. This class of afterimage tends to fade fairly quickly, but perhaps you got a sense of it. Now, I'm not sure if it happened for you. Perhaps you perceived the afterimage, whether or not you perceive where the colors were and what happened. And it may return. Yeah, some folks comment return. It may sort of flash strongly initially. It may sort of fade to white. And then if you keep staring, it may sort of come back. At this point, it's probably faded. The yellow, someone says, oh, the yellow ended up on the bottom. And if you recall my description, the yellow was on top at first. Yeah, so this is something that you may have perceived. You stared at this, and then after put into the white background, the afterimage looked like this. And this is the sense in which opponent color pairs or axes are meant. So blue and yellow is an opponent axis. It turns out red and cyan or red and greenish, reddish greenish axis is on the other side. Okay, let's take another example for fun. Okay, stare at this one. I'll do this for shorter. I have a few more examples I want to take you through. So if this one doesn't appear as strongly, that'd be fine, but let's give it maybe 15 seconds or so. You can tell apparently it's something like the American flag, but the colors are kind of off. There's green stripes instead of red and white, and there's a yellow background instead of blue. Okay, ready for flipping. Here we go in 3, 2, 1. Again, try not to move your eyes. All right. So if you're like me now, you might perceive very strong red and white stripes and a very strong blue background with maybe white stars in it, but perhaps they're too fuzzy to be able to perceive. Okay. All right. These are stronger. So try this. Stare at the dot in the center and sort of observe there's some coloration in pink and blue. I'll give this another 15 seconds. Yeah, this one's a fun one. So I encourage you to try to stare strongly and with a steady gaze at the center. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0. Here we go. Okay, so observe sort of some pink and blue areas, and then sort of what happens after. Okay. This one tends to persist for a lot longer because there's a lot more sort of detail structure in the black and white in order to allow your gaze to rest on and to keep your gaze steady, and then to allow the color to stick onto it. Yeah, it looked like it's totally in color. And after a while, if you move your eyes, it's like, okay, this picture is like totally black and white. So apparently these colors that were on the red axis or pink red axis, if you will, or and then the blue went to colors that were in the green and the yellow axis. Okay, last example before we leave afterimages. You can stare at this one at the black dot. Keep staring at the black dot. This one, while you stare at the black dot, I'll observe that the sort of upper area of this picture is pretty clearly yellow. The center part of the picture is, well, somehow I triggered it by accident. Okay, anyway, okay, you get the idea. Okay, so now we got a blue sky, we got green grass, we got yellowish brickwork. It's kind of weird. Didn't think I was advancing that, but somehow it auto-advanced. Okay, anyway, so you got the idea. Okay, so you can play around with these illusions at your leisure as well in the slides. But some of these are pretty striking. And again, the theme here was after images. These are illusions that have been crafted in order to illustrate the principle that the human vision system, in its coloration or its treatment of color somewhere in your brain or your neural processing, runs, I guess, across these antagonistic axes of color, where blue and yellow are on an antagonistic line, and red and green are on an antagonistic line. So if you stare at red for a long time and then turn off that red, then your visual system tends to see green as an after image, and similarly for blue and yellow. Okay. Yeah, if you haven't seen it before, kind of striking. Okay. Uh, Whenever I find the human vision system and the visual system is just so complex, so surprising. It's so weird. I want to share with you one that I learned relatively recently. That's one of my favorites. It's called the watercolor illusion. Okay, so take a look here. You might see what you see is a purple squiggle. Okay, that's making something that might look like a donut to you, sort of outlining a donut shape. Okay. Okay, so I'm going to show you the same picture, but here it is. And what I want you to observe is, okay, something happened with it. You can stare close at it if you want, to the line. It's still a purple line, but there's some kind of yellowish on the inside of the line. If you stare close, you might perceive that. So what I want you to do is pay attention to this square donut shape, if you will, and perhaps you perceive it more strongly as a shape, like a square donut shape than before, okay, than here. Here it was just an outline of or two purple squiggles. Perhaps here it feels a little bit more solid. And one of the ways you can sort of try to figure out what's going on there is to look at the color that's inside the hole of the donut, if you will, compared to the inside of the donut itself, compared to the outside of the donut. So let me ask you, you folks, for how many of you does the inside of this quote unquote donut, the whole of the donut, look exactly the same color as the donut itself? For how many of you does it look exactly the same, or does it look different? Different. A couple of people saying different. The donut is yellowish. Different, different, different. Looks more white. I think inside, but I think I can tell that they're the same. A lot of people saying different, more white in the center. Yeah. You know what it is. This stuff here is RGB white, okay? It's 111, exactly the same as the center. So the only thing that's different here is that this is a purple squiggle. This one over here is a purple line with a yellow line on the inside of it. That's it, okay? That is the only change. And yet somehow, for most people here, certainly for me, it's very clear. Like this donut here, I wouldn't call it yellow inside here, but it's definitely doesn't look white to me. Whereas the whole of the donut totally looks white to me. Okay, so what does it say? Well, I don't know exactly, but evidently the human visual system has some kind of flood fill feature, I guess, right? Where the color that's only on the periphery of this line, of this squiggle, is somehow my visual system is transporting it into the interior and flood filling this somehow to make it look a little bit off-white. What? Kind of crazy. Okay, so that's sort of the end of the fun grab bag of crazy, simple but crazy sort of illusions of showing different aspects of the human visual system, some of which we're not going to come back to, but some of which we will come back to when we talk about color encoding, because they turn out to be influential in setting up how to encode color spaces and things like that that are perceptually based. We'll come back to that towards the end of Thursday's lecture. But now, having sort of left the world of human visual system and color perception is highly adaptive, has sort of interesting weird features like chromatic adaptation. It's really surprising weird features like flood fill from the watercolor illusion. Having left that now, let me say, because it might sort of seem like, well, what are we going to do with color? Because it's so slippery, or human visual perception is so slippery. Let's leave that now and sort of center on what is true, and from an engineering perspective is really critical, is that make no mistake, we understand color reproduction, which is a critical, color processing, color engineering task, obviously. We understand color reproduction as a quantitative science, and it's really honed these days. We all know that, right? All the devices we use, the display that you're looking at this lecture on right now, your phone, when you take photos, the color fidelity to our experience in the real world for the kind of imagery that we see on our devices is very authentic. It's very convincing. Okay. And that's because we understand color reproduction as a quantitative science. Now, we know the displays, we're marketed to about this, is that the displays have tremendous color, rendering capabilities. The color gamut, again, the space of colors they can represent is really brilliant, rich, and getting better all the time. And we know that these devices really are remarkable visual objects. We'll talk about color gamuts of some of these things and how they're measured later, how we quantify them later. And we know if you haven't had the opportunity to take a magnifying glass, I encourage you to take it to your smartphone and just look. It's there's no mystery there. They're so small that you can't see them with the naked eye. But with a little bit of magnification, you can, you see clearly that they're just red, green, and blue dots, pixels. They light up to different amounts. So this is just black text on a white background. So the white background is just red, green, blue pixels illuminated, basically full brightness. And in the black text, it's the same red, green, blue pixels. They've just been turned off, right? The brightness is very low. And from our anti-aliasing, you won't be surprised that along the boundary here, there's intermediate intensity values representing, partial coverage or partial gray values. Okay, so we know that's the case. And then when you put it together and look, like as I was saying before, you look at a photo, right, on a modern device that's been taken in a reasonable way, and you're like, yeah, that color looks totally convincing. That looks like I'm quite sure like how the lavender, bushes looked in this picture, below that hill for the photographer that took this, right? The way that we do that is on a pixel by pixel reproduction level. We take each pixel and we sort of say, what is the color that we're trying to reproduce here that existed in the world? This is the color reproduction problem that we're going to study from a calculation perspective, the math, and then the computations involved in this. So to set it up, we'll come back to it later when we talk about it, but the problem is simple. Let's say I look in the real world, and I see something like, okay, a damselfly clutching onto a little stalk of a green stalk of a plant. And I say pixel by pixel in here, I could pick the green on the damselfly body, but let's say I pick the beige in the background. I say, I want to reproduce this visual appearance of this image on my computer, okay, on my computer screen. So how do I pick the pixel values for each pixel in this scene so that the displayed image of the damselfly matches the appearance of the colors in the real world? That's the color reproduction problem that we will study, and that is really, really worked out in detail. Okay. We'll come back to that in a little bit. But before that, we have to set up some of the pieces to be able to set up how to pose that problem correctly from a physics and a human observer, biophysics, and visual neural processing perspective to be able to set up the mathematics to pose that color reproduction problem technically so that we can go about solving it mathematically. Okay, so now we're going to transition in this lecture to talking about just a little bit about what is color, but then getting into some of like what is the physical basis of color, what is the biological basis of color in the human observer, things like that. Okay, so what is color? Actually, I was tempted to almost take out this slide, what is color? And it's really interesting question. It's a very natural question. What is color? But it's also an incredibly complicated question to answer. And I think one way to get into this is you say, well, what is, the experience of seeing redness, right? And, it's almost like if you haven't experienced it, then, then I can't really answer your question. You know what I mean? And that has to do, again, with sort of like the notion of color as an internal experience, a human perceptual experience that is very much in the mind. So a few comments about it, recognizing that this is one of the very slippery things that makes it difficult to define color in absence of context. And there's so many different contexts, from human to artistic to aesthetic to scientific to physics to chemistry to neuroscience from which to consider it. Nevertheless, let's take an attempt at some of the really big important features of it. Okay, so what is color? Okay, one of the really basic facts about it is that color is a phenomenon of human perception. Okay, it is not a universal property of light. There isn't like, oh, here's some light falling on something and it has that light falling on something has a color. It is not intrinsic to the light. It's a property of an observer looking at it and then forming an internal sensation of what it is. Okay, so colors are the visual sensations that arise from seeing light. This is starting to get a little technical. Seeing light of different spectral power distributions. We'll come back and define those spectral power distributions in detail if you've forgotten about exactly what I meant by that from the radiometry lecture. Okay, but again, colors are visual sensations. It's a phenomenon of human perception. It appears in the mind. It exists in the environment, but it's as much a property of what the observer is seeing as anything that is intrinsic. Now, one way to sort of heighten this is that, it's sort of easy to look at the world and assume that our visual experience is the truth, right? It's so convincing when we look at the world that this is the coloration of the world. But recognize, for example, that, there is no other animals that look at the world. There's no chance, and we could study this. I won't have the details of this in this lecture, but take it as a given, as a fact, okay, that there's no way they could perceive the world in the same way as us, because the color pigments in their retinas are not the same as ours. They may have a different number of those, independent number of those color pigments. So the dimensionality of their color experience may be fundamentally different. Or they may just have different characteristics that respond differently in different parts of the range. For example, you may have heard that bees are sensitive to ultraviolet light. As human beings, we're blind to ultraviolet light because we have no biophysical mechanism for receiving that light and sending it in to our brains. Okay. Someone asked, is color related to wavelength of light at all? It is. We'll talk about some of those characteristics, but again, as you might expect from this preamble, this extended preamble in this, in this week's lectures about the adaptive nature of light, the contextual experience of all of it, that it is not an intrinsic property of wavelength in and of itself. By the way, that's a very common misconception about light. Sometimes we think color is wavelength or wavelength is color. That's actually a fallacy. It's true under certain viewing conditions, sort of standard viewing conditions, if you will, but it is not a truism. It's not an intrinsic property of the wavelength of light in and of itself. Okay, we'll get maybe back to some of those details later. But let me explain this picture and these little terms down here in this slide, again, due to Mark Fairchild. So this picture you can see here is of a human observer, recognizing the fact that color, again, is something in the environment, for example, looking at these objects, these Crayola markers in this case, but what's arising in the person's mind in response to viewing this scene. And it comes from a starting point of having a source of radiation of energy, right, of a radiomagnetic energy in the visible spectrum that's going to strike some objects. That energy, that radiation energy is going to interact with those objects through physics, through chemistry, through the spatial geometric transportation of light, as we talked about, for example, in BRDFs and reflection functions. It's going to reflect off those towards the observer. The observer, in this case, this little girl's eyes, they come to the retina, and then there's neuroanatomy that's critical there on the retinal surface for receiving that reflected electromagnetic radiation and absorbing it in some kind of way, measuring it in some kind of way that we're going to talk about in detail. And those signals are sent to the brain, from which the brain, with all the psychological context, the other mysterious context that we've looked at some illusions today to get sort of a peek under the hood of, interprets that and then sort of gives us a sense of color. Okay, so in certain branches of color, it's really central to, or color science, it's sort of natural to ask, what is the color at the end of the day? Okay, is it orange? Is it red? Like what's the name of it? And then is it saturated? Is it a very intense red? Or is it a very sort of like pale red that's close to white because it's so pale? Is it really bright, like, brilliant, close to like a white sheet of paper? Or is it a dark red, like very close to black? Things like that. So sometimes in some branches of color, we attempt to try to, be able to quantify, to name those sorts of things. That's has to do with things called color appearance models. Actually, for our purposes in this lecture, this introduction to color science, we're going to center ourselves into really more measuring and quantifying light. And I'll touch on how we can get into color appearance models a little bit towards the end of next lecture in naming these colors, but we'll actually satisfy ourselves mostly in trying to reproduce colors, which won't require us necessarily to be able to name them. Anyway, in terms of what is color and the attendant, sort of scientific and technical disciplines related to it, you can see there's a whole bunch, right? So in the optical radiation sources, there's physics. We've talked a bit about this in terms of radiometry, characterization of objects, most salient in this case, as highlighted by Professor Fairchild, or the physics of the interaction of light and how they reflect off these objects, as well as the chemistry of related to at a wavelength by wavelength basis, how it sort of reflects. And then in the eye, anatomy, physiology, psychology, a very large range of topics that are, related to related to color science. Okay, so with that sort of a little bit of acknowledgement of the breadth of color, the difficulty in sort of even the basics of defining what we mean by color, let's get down to some of the facts. Okay, so let's sort of center ourselves into things that we can be sure about and start to talk about some of the facts related to it. So let's start by talking about the physical basis of color in electromagnetic radiation. So famous picture, very common, one that we've all seen before. You can separate white light, such as something that comes from the sun, from sunlight, for example, through a prism into a rainbow. And this is happening through dispersion, through things that we talked about, Snell's law and chromatic separation of light that we talked about in some of the lectures on material reflection. And this is related in the history of ideas, right? Scientific ideas. Again, color is such a fascinating topic. If you look in the history of science, okay, one of the giants, of course, is Isaac Newton. And if you think of the scientific method, actually one of the experiments that is pointed to as the prime example of the birth of the scientific method is the experiment sort of shown in this painting or depicted in this painting, where Isaac Newton separates light into a rainbow, exactly using that method shown before with the prism. This is often referred to as the experimentum crucis, or the crucial experiment. The gist of it is that Newton showed that you can separate sunlight, you can subdivide it into a rainbow with a prism. There's a few pieces in here. He has a prism here that he's sending light through this slit and then putting onto a cardboard here. You might notice there's another hole shown here. So he can, by moving where that rainbow occurs, he can choose which portion of which wavelength here that's been separated out and the attendant color through that hole, and send it through another prism. And one thing you find is that you can't further separate. Okay, Newton shows that sunlight can be subdivided into a rainbow with a prism, but the resulting light cannot be further subdivided. Okay, you can't take yellow light and then subdivide that into a rainbow as well. We'll come back to that a little bit on exactly what's going on there, but that's because it's been separated into individual wavelengths of light, at least is the typical scientific interpretation of that experiment. Okay, so what wavelengths are we talking about? This is, of course, the visible spectrum of light. Electromagnetic radiation, as you may recall from physics or from EE classes that you may have taken, is like a huge range from gamma rays and X-rays, UV, infrared, microwave, FM, AM, long radio waves. It's a huge spectrum of wavelengths and attendant energy for the photons associated with different classes of electromagnetic radiation, but they're all photons at the end of the day. For light and things that we can see as human beings, we only mean in the visible spectrum. Those are the only things that we can see, right, as humans. And the visible electromagnetic spectrum, rather, spans a range from roughly 400 to 700 nanometers or so, okay? So what's a way to sort of analyze some of those or separate those lights out? It's basically what Newton created. A modern version would be a device called a monochromator. So you might have a light source that you want to analyze. You put you create a beam of that light by passing it through, for example, a slit, pass it through a prism. The action of the prism is to separate out through dispersion into, so that the angle that exits is correlated with the wavelength of the light. And therefore you get a rainbow spectrum. Someone asked earlier, right? Is color related to wavelength of light at all? Yeah, so when it's separated out in this sort of a way, usually the long wavelengths, 700 nanometer, associated with red, the short wavelengths with blue, the intermediate wavelengths with green, orange, yellow. But this is under standard viewing conditions. So one thing we'll have to talk about later is, right, what happens after that light comes out, those wavelengths, like, so what goes into the human observer and what happens biologically. Let's come back to that. But let me just observe that the same wavelength of light under certain viewing conditions might look like very different colors. Okay, you might have gotten that sense already from chromatic adaptation or the discussion about that earlier in the introduction portion of this lecture. Anyway, okay, so this is a monochromator. You can control which wavelength finally exits the monochromator. In this case, we've selected a, say, 540 nanometer, 545 nanometer wavelength, which looks green under standard viewing conditions, by positioning the second slit so that it's at that portion of the wavelength spectrum. And you can control which wavelength exits by changing the angle of the prism. You can turn this into a measurement device just by adding a detector on the end and then systematically scanning over these wavelengths by changing the angle of the prism. So for an unknown light source, you can use the monochromator to isolate each wavelength one by one, and then use a detector to measure the associated power at that wavelength. If you do that, then you end up with, you could graph that as a function of wavelength, and that would be a spectral power distribution. Okay, so this is the salient property in measuring light, especially as it relates to color science and color perception. So it's just the amount of light present at each wavelength. The units are from radiometry, very much related to radiometry. So they're radiometric units per nanometer. For example, they could be watts per nanometer. It's also kind of common in considering spectral power distributions in color science for it to be unitless. And then they just sort of, for them to be unitless because we don't necessarily care whether it's the irradiance that's falling on a surface or whether it's emitting from a light source. We just sort of care about the quality of the light itself. And in that case, often it's common as well to use relative units when the absolute numbers don't matter. We just use the relative amounts at different wavelengths, scaled to, for example, the maximum power at the maximum wavelength, so that we can compare the relative power across wavelengths. Let me show you an example of that. So in this case, right, this is just relative power. We don't know what the absolute power is at all. We should have to define how big the detector is or the portion of viewing angle that we care about. And that doesn't really matter in this sort of scenario where we're just trying to say that, hey, the blue sky has a spectral power distribution. Again, this is a power as a function or a variation of the wavelength on the horizontal axis that you can see for a blue sky spans a whole bunch of different wavelengths here relatively uniformly, and certainly uniformly compared to like the solar disk. So if you think of sunlight itself, it's much higher energy towards longer wavelengths. It rises towards higher wavelengths, okay? So these are some examples of daylight spectral power distributions, or SPDs. Here are a few other examples of SPDs, again, for daylight, incandescent, fluorescent, halogen. Here's an LED example. Here's a warm white LED. For fluorescent, for example, you might notice it's really spiky. This has to do with the physics of how the photons get emitted because of the way that we excite the atoms that are present inside the fluorescent tube. And then when they decay in sort of the energy and they fall to lower energy levels, that jump in energy is emitted according to the electronic structure of those atoms at very specific energies, and those energies are specific to the wavelengths that show up in the spectrum. So different light sources can have spiky SPDs, smooth SPDs, or peaked in certain areas depending on the underlying physics of the creation of the light source. Okay. The next property of SPDs I want to highlight is linearity or superposition. So light, again, we talked about it in a lot of the ray tracing and radiometry things that we have looked at in the past, and it totally applies here as well in SPDs because it's just across different wavelengths. Things add up linearly when you combine them. So for example, you put this light source here that's colored bluish into this spectral radiometer, and you got this SPD measurement. And then you had a sort of yellowish one, you put it in, you got this power measurement. If you put them both in, it's drawn a little crudely as a cartoon here, you sort of have to type them both in so that they're not going to get blocked, obviously, by the opening. But if you manage to put all of the light into this device together, then you would get the strict superposition of these two SPDs over here. Okay, so the SPDs would sum point by point or wavelength by wavelength in linear proportion. Okay, so that's some stuff about the physical basis of light as it's going to relate to, or the physical characteristics of light as it's going to relate to color. Let's switch gears and talk a bit about measuring light. We already talked a bit about measuring light in an SPD sense, a spectral power distribution sense, where we carved out each wavelength. In this case, what I mean by measuring light is without using a spectral radiometer, which is a pretty specialized device that we don't have access to. In this case, we're going to think of measuring light in a general case, but where it's going is you can think of this as occurring in a cell on your retina, which you can think of as like quote unquote a pixel on your retina, or literally in a camera as one of the pixels on the surface of a CMOS sensor or something like that. Okay, so here's a simple model of a light detector. It produces, it's a device, it's a small component or device that produces a scalar value when photons land on it. Okay, so a scalar value, like literally a number. The value depends only on the number of photons that are detected, and each photon that lands on the detector has some probability of being detected, okay? And the probability of being detected is going to depend on the wavelength. It depends on the physical characteristics of the detector, but for any specific detector, it's going to vary. The probability that it detects a photon will vary with the specific wavelength. Now, this is a really crucial point, okay, in this model of a light detector, is that for the detector, there's no way to distinguish between signals that are caused by light of different wavelengths, okay? There's just a total number that comes out of the detector. So the detector is, again, there's photons that arrive on it, some fractional likelihood that for each photon that arrives, it'll get detected, and then a sum total of how many photons got detected. But the detector has no way to tell whether it was at one wavelength or another. It can just tell you how many it managed to detect. That means it's going to be blind to wavelength, okay, which is a really crucial point in what it can or cannot tell us. Now, it turns out this is a very simple model, but it works for many detectors. It works for detectors based on semiconductors. For example, I said pixels in a digital camera. Turns out it's going to be a good basis for what's going on in the photoreceptors in our eye, where the interaction of this kind of detection is happening in the visual photopigments that are present inside, the cells in on our retina. And this is a cartoon that shows what I said on the previous slide. So to anchor it, okay, so what's the model again here? Here's the detector. Here's like a rain of photons that is falling on the detector. In this cartoon, the different wavelengths are depicted by different colors, okay, of these photons. That just represents that they come at different wavelengths. Now, the incident photons will have some kind of distribution. Okay, so the horizontal axis here is wavelength, 400 to 700 nanometer. So the vertical axis for this here is the number of incident photons. Okay, so there's some, as a function of wavelength, there's some distribution of how many photons are arriving at the detector of that wavelength. That's the signal. Then there's the property of the detector. As I said on the previous slide, any specific detector will be able to measure some wavelengths. It's going to be more sensitive to some wavelengths than others. Very typically, it's some kind of a bell curve. Okay, in this case, it's most sensitive to like a middle visible wavelengths, call it 550 nanometer or something. There's a peak here, okay, in the detection efficiency or the sensitivity of this particular detector at 550, and then it falls off in other directions. If you had a different detector, that peak wavelength might be centered somewhere else, right? Of course. So that's going to be a specific property of the detector. Anyway, given these two things, the signal, the spectral power distribution, this is not quite a power, this is incident number of photons, but they're directly related. The distribution of photons and the detection efficiency, we can calculate exactly what the signal that's going to come out is. We just multiply, right, on a point-by-point basis for every wavelength, how many photons arrive times the probability that they were detected, and then we sum it all up. So it's the area under the curve of the multiplication of the two top curves, as shown here. Okay, so the purple area here is going to be the signal that comes out of this detector. Now, again, just to draw the point that I made on the previous slide, if the distribution at the top was all really short wavelength photons, so it's just peaked here, you would get some area under the curve and it would come out. You could have another signal that came in later that was just all really long wavelengths, but just happened to have exactly the same area under the curve, and you would get the same exact signal number out. There would be no way for the detector to tell you that in the first case, it was short wavelength photons, and in the second case, it was long wavelength photons, That is not information that this kind of detector can provide us. Okay, really crucial point again about what it can and it cannot tell us. Okay, so just a little bit here. The same math on the previous slide carries over to spectral power distributions as well. That was for number of photons per wavelength. Spectral power distribution just says the power at a specific wavelength. You may remember from physics that every photon, based on its frequency, carries a certain amount of energy, right? It's related by Planck's constant. Anyway, so every photon of a specific wavelength has a specific packet amount of energy. So the math on the previous slide just carries over to spectral power distributions by just converting from number of photons as a function of time through to what the attendant wattage would be. The light entering the detector has its spectral power distribution S of lambda, and the detector has some kind of spectral sensitivity or spectral response R of lambda. Again, this is the likelihood of detection. So the measured signal X then will just be equal to the input spectrum S of lambda times the detector sensitivity R of lambda, is integrated wavelength by wavelength over all wavelengths in the visible spectrum, okay, or the detector sensitivity spectrum. We'll be using this later. I'm just developing this math a little bit, which we'll be using a lot in next lecture, just what this looks like. So this is an integral version of this equation. You're going to write it in dot product form. If S, the signal, that's the function of the spectral power distribution as a function of wavelength. If we just write it as a vector, right, where each cell in the vector just represents how much power at a sort of specific discretization, wavelength by wavelength, of the light source, we can write it as two vectors, and then S dot R, a dot product between those vectors, is going to give us the same X. We could also write it in matrix form if we wanted, with a column vector, say for R, and a row vector for S. All of these just different ways of writing the same mathematics that's represented by this summation of area under the curve, where it's amount of photons per wavelength multiplied by the sensitivity of the detector at that wavelength. Okay, all right. Now, I sort of belabored this point a little bit, but it's really crucial in understanding color perception and how limited color perception is in terms of what spectral information it provides to our brains and our understanding of the world. Let's start with this detector case, and later we'll look at what it is for a human color perception case. But in this simple model of a detector, recognize that we have a dimensionality reduction from infinity to 1. It's a huge reduction in information content. What do I mean by this? The spectral power distribution is a function of wavelength. A function of wavelength has infinite degrees of freedom, right? Every position or every wavelength in a continuous sense could have a different power. Or if you think more in terms of sort of a functional or mathematical functional analysis point of view, you can think of it sort of as a Hilbert space or expand a function in a Hilbert space, and that's an infinite dimensional or countably infinite dimensional linear space. So the spectral power distribution has infinite degrees of freedom. The detector result is a scalar result. Okay, so it has it's a one-dimensional signal, has 1 degree of freedom, just what the scalar output value is. So we take this, incredibly complex, potentially incredibly complex input object, the spectral power distribution, a function of wavelength, and we collapse it all down to one detector signal, which is what's the net total of signal that was detected by this specific detector class. Okay, let me see how we're doing on time, and let me see what might be a good place to pause. Okay, let me just tee this up and then we'll talk about color matching in detail next time. Now we're going to start talking about, that was, okay, a bit of physics, the physical basis of light as it relates to color perception or to color science, and then a bit about detectors and this simple model of light detection. Now we're going to apply that simple model of light detection and start to build it up towards what is going on in the human retina, where it turns out we're not going to have one detector. It turns out we're going to have 3 detector classes because we have 3 different types of color-related cells. That's sort of where we're headed. Hence, tri-stimulus theory of color, representing that fundamental nature of why our vision is 3-dimensional, coming from 3. But before getting into sort of the biological basis and what's been understood by a lot of intervening science, decades of intervening science to bring clarity to this situation, I want to take us back to the beginning and sort of say, how was, from a theoretical and well-grounded perspective, how did we figure out that color is or human color perception is three-dimensional? And this goes back to the very famous so-called color matching experiment that lies at the foundations, is really the bedrock of colorimetry, measuring color. And I put this description here, searching for a linear systems basis for color. Okay, so we'll talk about this in detail next time, but let me tell you about the historical experiment. There's just one slide on this. This goes back to someone very famous, okay, James Clerk Maxwell. Maxwell did a lot of really amazing things in engineering and science. Of course, he's perhaps most famous for the electromagnetic equations, unifying the electromagnetic equations. But before he did that, some of his earliest work as a scientist was in color theory, believe it or not. And he was the person that laid the technical foundations for colorimetry based on this color matching experiment. The experiment is very simple. He takes this spinning top, as you can see here, you hold it on the handle, and then you can put colored pieces of paper, different area sectors there, red, green, blue as an example on the outside, and in the middle, in this example, black and white to a certain degree. And what you do is you spin it, okay? So it's a metal disc plate that can spin. You can spin it really fast, and if you hold it in good light, the spinning colors will blur together, and you'll see the average superposition, linear superposition of red, green, blue in this proportion. You'll see that color, and in the inner disc, you'll see a gray that's mostly close to black, okay? And the color matching experiment is to just play around with how do you find the proportions of color, red, green, blue, for example, that can match different test colors in the middle of this annulus. This was the simple experiment that laid the foundations for how to measure color in a completely systematic and precise way, and that we'll talk about in detail beginning next lecture. Okay. All right, folks, we'll pause there for now. Thanks.